books. Here we go. All right, so what have we got going on this week? We'll have a quiz on Friday. There'll be I've got office hours this week are Monday and Wednesday, 1:15 to 3:15. So since we don't have lab officially this week, since you're coming in on your own, I can do those extra office hours if you want to come in for help. I'm not going to do the Friday office hours because nobody's probably going to come the afternoon before spring break, right? And then a Thursday help session, usual thing. Blah, blah, that's at 8:30 for you guys. Okay. Questions on stuff, and then we got lab stuff. Okay, so I have the lab stuff upstairs, though I haven't opened it up and fully set it up, but kind of ready to go. Let's talk about lab the next 2 weeks. I actually don't know what numbers we're on. Do you guys know? 6 and 7? Is it 6 and 7? Okay, so do 6 and 7 on your own.
That's 3 weeks, right?
Yes. So if you want to come in over break, if you're local or something, or going to be around, I can help out with that, but I need to try and get everybody on similar days, okay, so I can have things open. So send me an email, let me know about that if you're going to come in over break. Let's talk about 6. 6 is 3 days. It's the worst of those 3 days. The good news is it's not really a whole lot of your actual time or your time and attention. So the first day, you're going to start a culture. Start a 10 mil culture. All right. The antibiotics we're going to be using, it probably says kanamycin is spectinomycin in the manual. So you're going to use chloramphenicol, and I'll have these out, right? I won't have spectinomycin out. Instead of spectinomycin. All right, you guys are using Rosetta cells instead of the kind of more generic BL21 cells that we used to use. It's just they're different cells that we had for them. So you're going to start that culture. Run overnight. Just one night. Okay, you can't let it run multiple days and grab it because the bacteria will run out of resources and they'll all start dying, and it'll make the next day take a lot longer. It would still work, but it's like when I've left my my starter culture, or not starter culture, wrong. My oh my God, what do you call it for your sourdough? Your starter, okay, so it is starter. But my yeast starter, when I've left that in the fridge for 3 weeks and not made bread, the next time I pull it out, it's really slow getting going. Okay, it's been sitting too long since it's been fed. So you're going to start that culture. It's going to go overnight. The next day, second day, which is the next day, add 1 milliliter of starter culture, of 10 mil culture. And 1 milliliter, not 1 microliter, is a mistake sometimes people make. If you start with 1,000 times less, it's going to take a lot longer, right? Okay, so 1 milliliter culture to 100 milliliter flask. Okay, you're gonna add your antibiotics in this, like it talks about in the directions. Shake. Once again, chloramphenicol instead of spectinomycin. Shake. At, does it say, it says say room temperature overnight. And that's on that, there's a floor shaker upstairs for that, right? It'll be close, it's near the window. I'll pull it out a little bit. It's gonna stay on the floor because it likes to walk around. And when it walks around and off a table and falls on top of the flask, it's messy. And yes, that has happened. Okay, third day. Centrifuge cells. Centrifuge. Pour off supernatant. Put in the -80 freezer. Don't, and that's going to be the right one, meaning both the one on the right and the correct one. Questions? You're going to centrifuge those cells down in Dr. Hughes's lab with his centrifuge down here. Okay. All right. So in terms of the timing for that, I'm going to work to make sure those labs are open for you. When you do this centrifugation, remember that Dr. Hughes wants you to balance these things to, what was it, half a gram? So you're going to need to balance that to half a gram. Let's go ahead and talk about that. I should ask the department to buy another one for us so we can treat it badly. And then you're going to pour that stuff off. Pour it off into bleach. Okay. This should be in the directions in terms of that. Questions? Okay. And then the next set of things is you're going to do freeze thaw. So 7 is freeze thaw. So this is the worst because you have to come in, you have to find a little bit of time on this first day. You have to have a bunch of time on this second day where you're kind of available. So you're going to start this culture. You're going to wait 4 hours. Maybe write that down. I don't know what it says in the lab manual. Wait 4 hours for your first check, but it may take 6 or 7 hours. All right, so this is like a start in the morning, come in and check it sort of thing. And you need to catch it in terms of the absorbance between 0.8 and 1. You don't want to go above 1. Okay, that makes sense? All right. And then third day, you need to come in and find some time. It'll be 15 minutes to do this. So this is a bunch of kind of waiting time, but you can be doing whatever you want for almost all of it. Okay. Second thing is freeze thaw. There are 3 freeze thaw cycles. All right, so you're going to freeze in liquid nitrogen for 5 minutes, and then you put it in ice and let it thaw until the pellet's kind of swishy around. You might want to tap it on the bench some once it's started to thaw, not when it's frozen with liquid nitrogen. That's a good way to have something shatter, right? You've seen the freeze the banana, not the banana, but the roses. The banana you used to pound a nail in, right, to a board after it's frozen. But you can stop. This is more flexible. Can stop after any freeze or thaw. So what I can do is I can freeze my cells and I can put them in the freezer again, the -80 freezer again, and then come back a couple days. Nothing bad's going to happen there. Or I can have finished the thaw and put it in the -80 freezer. If I put them in the -80 freezer after a thaw, the next time I come out, I don't have to freeze them in liquid nitrogen. I just pull them out and put them in the ice. Does that make sense? Okay, but that's more flexible. You could spread that out over, I don't know, probably a year if you wanted to, but you won't finish lab. Okay. So for this, when we get here, if you're the first person doing this, you could do all this week. Okay, but I need to know so I have liquid nitrogen available for you. Because liquid nitrogen's down with the NMR. I don't even know if my key works for the new NMR, but I'll need to go get it. Your keys won't, if you have keys. Yes.
So you said it was 5 minutes in the liquid nitrogen, and then what's the?
It's like, so it's until the cells move around. That first thaw is awful. It's like 2 hours, 2.5 hours. But you just stick it in ice. You go do whatever you want. You come back maybe in an hour. You tap them around a little bit. You go away again. And then the next 2 thaws go a lot faster. Something to do with the cell structure or something. I don't know. That's just the way it works. Okay. And for that, you can leave them thawing on ice for a couple hours for each one. I wouldn't leave it all day, but it's once again not super picky. Okay. And then at the end of that freeze thaw, I've got my final thaw. I add some PBS buffer to it and I let it shake on the shaker for 45 minutes. That needs to be 45 minutes on the shaker. And I'll set some stuff up in terms of uh, styrofoam container wedged into the shaker. So it's a gentle kind of shake. Hopefully we won't have too much overlap between people doing their various shakes. But we'll set that and be a deep thing to put your tubes in. Does that make sense? So what you shouldn't do, if you end up setting this up and I'm not around to ask questions or anything about that, if you're putting something on the shaker, don't take your freeze thaw tubes, put them in a beaker, put them in there, put some ice in like this, you put your tube in like that, and shake it really hard and go away for 45 minutes. Because what you're going to do is you're going to find this tube someplace else in the room. Does that make sense? Okay, so it's a slow shake, and I'll give you a big container of ice. I'll have that set, certainly once the rest of the class gets there. Questions? Okay, lots of details in the manual, just follow that. And really the only changes I'm thinking of right now is we're going to be chloramphenicol instead of spectinomycin. Okay. I don't know why I have beta oxidation. We're going to come back to it, but what we're going to talk about today is fatty acid synthesis. All right. So what this is a picture of here is this is Mycobacterium tuberculosis. I might actually do some computational research on this if I can ever find the time. Right now the computational stuff is more basic on improving the systems. But this is the causative agent for tuberculosis. In fact, one of the diagnostic tests of this is you'll take a blood sample, or actually no, it's a sputum sample is what you take. If you're worried somebody has active tuberculosis, what you would do is you take a coughing up sample, and then you look at them under a microscope and you're looking for the bacteria. Not perfect, you don't always see it, but that's one of the things they'll do in certain countries. Okay, 1/3 of the world is infected with this bacterium. Okay. Okay, it's an interesting disease because it has an active form and a latent form. All right, so in the active form, that's when somebody's really sick. Okay, this is the form that kills you. In the latent forms, people can just carry it and they're okay. All right, while they're carrying it like that. And in here, you've got like 10 million people have the active form. All right, and that leads to about I don't remember what year this was, but it won't have changed much. 1.3 million deaths from tuberculosis each year. All right. I think there's a vaccine now, but if you're living in there's a real disparity in terms of being able to get medical care and get treatment for tuberculosis and be able to get the drugs in the third world or other developing countries, I guess I should say. Okay. If you're interested in learning more about this, who knows? Anybody know who Hank Green is?
It was his brother who wrote it.
Oh, I'm sorry. Yeah, John Green. Sorry, John Green wrote Everything is Tuberculosis. It's a great book. I got it. I think I got it audiobook from the library, the Salem Library, and just listened to it. So I recommend that. So it's a great story. I mean, it's a depressing story as well, but he writes a good book. All right, so in terms of treatment for this, so the treatment is 6 months of antibiotics or longer if it's disease or it's antibiotic resistant. All right, so think about that. You get sick and you get prescribed a Z-Pack for antibiotics and you take it for about 5 days and you're feeling better. And I don't know about you guys, but I sometimes have a hard time remembering to take the rest of it. I'm feeling better. I got this thing I've got to take. It might be easier. It might be easier if you're taking things all the time. Okay. But the treatment's 6 months of antibiotics. You'd have to do that for 6 months. And for a lot of that, you might feel like you're not sick anymore. Okay, but you do need to clear it from your system. And let's say now we make it, you're in a developing country and you have to take off time from work to go to a clinic to get this administered. Okay, even though you're taking them, they have to take it, the doctor has to watch that you've taken it. There's all sorts of things that make this more difficult if you're in a developing country. So you could imagine this is pretty tough, right? You get malaria, they give you 7 days of antibiotic and you're over your malaria. Okay, so there's also, this is really stands out in terms of this, and part of why that it's this long treatment, it's very slow growing. So that's one thing. It's hard to kill something by stopping it from making things that are essential if it's not growing very fast. That's how a lot of our sorts of things, our drugs work. And one characteristic of tuberculosis, and that's going to be important for our discussion today, because we're talking about fatty acid synthesis, it is very, very thick cell walls. With long fatty acid chains. Okay, and these specific fatty acid chains are called mycolic acids. They have some modifications to them as well. Okay, so here's an example of the cell wall. If we look at the cell wall of any of the mycobacterium, okay? So what you've got is you've got your membrane, right? And you've got your peptidoglycan layer. We're used to that, right? We remember that from last term. And then we've got this arabinogalactan thing, okay, which still isn't our cell. And then on the outside, we've got these super long mycolic acids. All right, and these are important for the structure of the cell wall and for protecting the mycobacterium. Now, cell wall synthesis is a target of the synthesis of these mycolic acids is the target of some of the first line drugs for tuberculosis, including, this is isoniazid. It looks like this. Okay. Okay, this is a drug. That's first line drug for treatment okay, and it targets fatty acid synthesis. All right, so when we talk about, we've got something, got the fancy name, beta oxidation, right? The reverse pathway is fatty acid synthesis. Sometimes it gets abbreviated FAS, okay? And if we think about tuberculosis, so you have fatty acid synthesis 1, and this is synthesis of fatty acids from 4 to 20 carbons in length. Okay. Why does it start with just 4? What do you think?
Genetically favorable.
Yeah, you've got 2 acetyl groups, right? You bring them together, you've got a 4-carbon thing. Okay, so it's going to you don't make a 2-carbon fat because that's acetyl acetates, your acetyl CoA. Okay, and then we have we have that, and tuberculosis has that one. But they have to make these really long fatty acids, the mycolic acids, and so they have another system, fatty acid synthesis 2, which is the same pathway, but it uses different enzymes. And this is going to be synthesis of fatty acids. We're going to go 20 to, is it 60? Yeah, 60 carbons.
Wait, so why is it like, what is the 1 and 2 mean of it?
So the one, this is a set of enzymes. It's the same pathway, but this is it's like when we talked about beta oxidation and we had short, medium, and long, right? Same sort of thing here. We have kind of short to medium, I guess, but it's all and then we have long. But we don't do this, but tuberculosis does. Okay, and isonazid is going to inhibit this pathway. I forget exactly which step. It's one of the NADPH steps. It's actually an indirect. It inhibits something that participates with that step, but I forget the exact mechanism. Okay. Questions? All right. So you've got a bunch of drugs that are targeted at this sort of thing. This is what, just in case you were interested in what the mycolic acids look like in non-cartoon form. Okay, and so you've got these super long fatty acids, and then you also have some of this extra stuff going on, right? That's a long fatty acid. We've got an acid chain there, and we have 2 chains coming off. We add some of these methoxy groups sometimes. Sometimes we do our fun triangle, right? Triangle things. Questions so far? All right, so we're going to talk about fatty acid synthesis now then. What was that? The triangles, that's an actual, what do you call it? It's a, what was that?
It's an epoxide?
Well, it's an epoxide except that it doesn't have the O in it, right? That's just a carbon, carbon, carbon. That's actually, this is line angle. It's not cartoon at all.
Isn't that so like energetic? Like,
It is.
It's awful.
It's weird, right?
How does that exist?
It does.
Yeah.
I mean, you can make it apparently, right? So what have you done? You've taken something that would have been 109 degrees and you've made it 60. Yeah? It's super strained, but why? It does. I don't know what the function is in the membranes. Okay, fatty acid synthesis. I guess I should have printed this out for you. I did not get a chance to do that. This is, I'm going to scoot it way down. I'm going to make it nice and small so we can look at the whole thing. But this is essentially the 4 steps of beta oxidation, but in reverse, with some slight differences. All right, so what we do here, I'm going to point at stuff you can't see very well. I've got acetyl CoA. I've got a chloro fatty acid chain. I do my fatty, I stick the 2 together. And once I've stuck the 2 together, I've got 2 carbonyls apart, right? Remember that's the last step before we in beta oxidation. And then I go ahead and I'm going to reduce it, I'm going to reduce it, I'm going to reduce it until I get rid of one of those C double bond O's. And then once I've done that, I'll go back up again and I'll add another acetyl-CoA. Does that make sense? All right, so let's walk our way through that. in a larger form where we can actually see what's going on. All right, so you guys see this? So an important difference here between beta oxidation and fatty acid synthesis is that I said, hey, we've got this growing chain, but really what we've got here is what's called, this is the growing chain. Okay, and this is what we're adding. This is a malonyl-CoA. It's a 3-carbon thing with a carbon dioxide thing that can come off of it. All right, and it takes, and we'll talk about how it gets made, but for now, it takes 1 ATP equivalent, be in there, to make this from acetyl-CoA. Does that make sense? So we've got lots of acetyl-CoA, right? Remember, we've got our catabolism is convergent. We're going to make acetyl-CoAs, and then we'll either make them into energy, but we could use them to build up things like fatty acids. But for them to actually work in this, they need to be activated by putting a carbon dioxide on, and that costs me an ATP. All right, we'll talk about that specifically. What's the ATP probably doing? That we've seen over and over and over again. It's a coupled reaction. More specifically, you could phosphorylate. And what would be the point of that? I don't remember whether I'm gonna have to see when we get to my notes whether it's exactly phosphorylate or not. But what's the deal? We're giving it a good leaving group, right? We're gonna put if any piece, I think it's phosphorylate, but any piece you stick on one of these things, right? You stick an AMP on, you stick a phosphate on, you're putting on a good leaving group. Unless it's staying there, it's a good leaving group to help us couple to, that's how the reaction is coupled. Anyway, so we make this malonyl-CoA. Looking at this, so we've had things that are CoAs before, right? We've got, in terms of these diagrams, this is coupled to this is part of a big protein complex. So this is called acyl carrier protein. All right. So in other words, our malonyl-CoA gets stuck to a protein before it's going to get transferred in. And this is another protein that's part of the fatty acid synthesis complex.
Wait, so how was that one made?
I'm sorry?
How was that one made?
This right here? So this is we've got a protein complex. There's a cysteine, right? Then the cysteines onto a whole there's a protein. Okay, so let's do it. So maybe to highlight that, you guys have seen me do this before, right? Okay. And so we've got this in this fatty acid complex. And so what happens is we've got this CoA. I'm sorry, it's not a CoA. We've got the growing fatty acid chain is attached here, and it gets attached through just this reaction. Did that help, or not sure what the question was exactly?
I was confused, like between from the SACP and that one, and like.
They're both proteins, and the SACP is a sulfur on an acyl carrier protein.
Okay.
It's probably a cysteine as well.
Okay.
Right. I don't think it's a methionine. But I don't know, and it's not actually really important that we know this, except that you know that this is all part of a complex. And part of what is going on here is that, having done a little bit of research on this in the lab, is fatty acids are really not very soluble at all. They're a bigger pain in the asses to work with in the lab, but you can't get these things into solution at good concentrations because they're not very soluble. And And so, so these things get attached to these big proteins, and they're kind of sequestered inside from water, so you can actually do the work without having stuff crash out of solution. Anyway, just know that these are proteins. If you get asked what ACP is, know that it's acyl carrier protein. I don't know that I'm going to ask you to generate it from whole cloth. Okay. Once again, we need to know the intermediates and the reactions happening, but really they're just the reverse with some small exceptions. And you don't need to know the protein names or the enzyme names. All right. So moving backwards, then we have this malonyl CoA comes in. Okay, and we're gonna stick our growing protein onto it, right? Remember how when we do beta oxidation, right? We got this on a CoA. Which end of the fatty acid do we cut things off of? Is it 2 at a time from this end or 2 at a time from this end? It's this end, right? We go, that's 2, cut that off, then they'll move up and cut that, cut that, cut that. When we build it, same sort of thing. We're building from this end, build, 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 but we're building onto that part. So when you look at this, right, this is our growing fatty acid chain, but I'm doing this with my hand, but I really should be doing this with my hand, right? You're going to stick 2 on, and then 2 more on, and then 2 more on, like that. All right, so that's what you see here. The malonyl CoA comes in, it's activated, it's got this carbon dioxide. The carbon dioxide leaves, and now we're here, right? So last step of fatty acid or beta oxidation. So now we need to reduce this to a hydroxide. And remember, we use an NADPH for that. Where do we get our NADPH from in animals?
The pentose phosphate.
Pentose phosphate pathway. Okay, that's where this is. So when we talk about synthesis, being used in synthesis, this is an example of that. All right, so NADPH gets used to reduce this to a hydroxyl, right? And then for beta oxidation, we got the hydroxyl by adding water across the double bond. For fatty acid synthesis, the water leaves and we get the double bond. Okay, and that's what you see. I can scoot it up a little bit. And that's what you see here. Water leaves, we get our double bond. Okay, and then we just need to reduce that a little bit more. Well, that's a lot of energy, but a little bit more to get to here. And now we've added 2 more carbons onto our fatty acid chain. So we go ahead and use this again. Okay, and now we have a fatty acid. In this case, they just started with 2 and they went to butyryl. So butyryl's 4, right? You remember from organic chemistry. But you just added 2 on. You go up, do the cycle again, then you have 6. Do the cycle again, then you'll have 8, right? Okay, so that's the process we're looking at. And then in terms of the rest of this figure, I think they just hydrolyze it off. Right, it's attached to a protein. They add water, cells, are going to go ahead and water gets added across here when it comes off. Does that make sense? Okay. So that's an example. And here, I guess we were making is that a 15 or a 13? I think that's a 13. So this would be a 16 carbon fatty acid. I don't know the names of these, and I don't expect you to know the common names of them. Maybe showing up on your MCAT, I'm not sure.
Some of them, but not enough for it to be worth it.
Mm-hmm.
So what do we need to know from this figure, just like the general process?
So it's the same. What do you need to know from the figure? So you need to be able to do a compare and contrast between fatty acid synthesis and beta oxidation. You need to be able to, I could block out a step and you'd be able to fill that step in.
Like the
Draw all atoms. So I would block out this, let's say. that a good one? Yeah, that's a good one. I block out this. I might block out this and this. And so you need to know that, okay, I've got that. I'm going to reduce here. I'm using NADPH.
But we don't need to know the enzymes.
You do not need to know the enzyme names. You do not need to be able to draw it full from scratch. But you should kind of know, I mean, there's the, for beta oxidation, you've got your fatty acid, right? You need to oxidize all the way to a double bond, C double bond. So you start out, you put a double bond in, you add water across the double bond, you oxidize the hydroxyl to make the C double bond H, or C double bond O, and then you break the two apart. And for fatty acid synthesis, it's the opposite, okay? Except there's some energy differences here. One of our things, to add 2 carbons on, we come in with a malonyl-CoA, which is a 3-carbon thing, and we knock off one of the carbons. That's another difference, okay? And then we use an NADPH, which is energetically equivalent to an NADH, and we use an NADPH again. How is that different from beta oxidation? In beta oxidation, we used an NADH and an FADH2. We didn't use, we got them out. So you see it's more expensive to build this up than it is to tear it down. And that's almost always the case in biochemistry. When we look at these pathways, you have 2 opposite pathways, just like glycolysis and gluconeogenesis, where it was more expensive to make glucose than to break down glucose. Same thing here. Okay. And we're going to go ahead and count those now. So what I want to do is I don't think it's down here. Oh, okay. So you could get out your beta oxidation sheet if you want. I'm going to make this a little bit smaller, maybe. Actually, I'm going to assume we know this pretty well, right? We've got, for beta oxidation then, right? We've got, for every 2 acetyl group I pull off, I get an FADH2 out, which then becomes a ubiquinol, and I get an NADH out. Okay. So let's compare costs. Costs of beta oxidation and fatty acid synthesis. There we go. Okay, so we're going to consider an acetyl unit. Well, let's just consider 1 round, okay? So 1 2-carbon unit. If we're thinking about beta oxidation, and let's go ahead, PO ratios are 2.5 and 1.5 for NADH, NADPH. Okay, they're energetically equivalent, and for ubiquinol. Okay, those numbers. So beta oxidation, 1 round, we do 1 QH2, right? And we do 1 NADH. And so that's going to be 1.5 ATP for one of those and one of those. And then for NADH is going to be 2.5 ATP. And so that's going to be a total of 4 ATP. So that's what it costs. That's not what it costs, but that's the energy that you get out of that. And then there's more energy if you take that acetyl group out down and oxidize it. But we're not worrying about that. We're just thinking about if we were to run these 2 pathways at the same time. Let's go ahead and look at fatty acid synthesis. For fatty acid synthesis, we have 2 NADPH that we use. So this is yielded, and we're going to be using down here. That's times 2.5 ATP each, so that's going to be 5 ATP. Okay, plus, where else did we have ATP in that process? Make the malonyl-CoA, right, plus 1 ATP for malonyl-CoA. Okay, and so that's going to be equal to 6.0 ATP used. All right, so do you see if we were to run both of these pathways at the same time, we would be going nowhere, right? And we would be losing 2 ATP per cycle. What do we call that again if we run both of them at the same time?
Plus, there's a specific word.
Futile cycle.
Yeah.
Right.
It's close. I got the guess. It was like right there.
Yeah, futile cycle. So maybe I haven't used it in class. I'm glad I haven't.
No, you did.
Okay, futile cycle. When 2 opposite pathways run at the same time. Okay. Energy is almost always lost when this happens. Okay, we'll see when we get to ketone bodies, that's actually an example where the one pathway and the other pathway have the same energetic costs. It's probably driven by the fact that you make ketone bodies, they go to a different tissue and they're used there. So a Le Chatelier's principle thing. And of course you lose the energy of shipping the stuff around your blood, right? Your heart has to pump. Okay, so let's talk about the synthesis, or other questions about that? Sophia?
I just wanted to ask, like, if you could jot down like the differences that you said.
Okay, let's do that. So differences between beta oxidation and fatty acid synthesis. Besides that, they're pathways that run in opposite directions, the same pathway running in opposite directions. And I'm sure if we had an expert in the field of either of these, they'd be offended by that concept, that statement. Okay, but what we have to do is beta oxidation, fatty acid synthesis uses malonyl-CoA, right? So I'm going to say fatty acid synthesis adds a malonyl-CoA or a malonyl group, or no, let's say uses. And I'm going to just emphasize here that's 3 carbons. 3 carbons costs an ATP to make from acetyl-CoA. To add 2 carbons to a growing fatty acid chain. Beta oxidation yields an acetyl-CoA, 2 carbons. So in other words, you couldn't just take the products from beta oxidation and immediately shove them into fatty acid synthesis, right? Outside of the first 2 carbon that you stick on the protein, on the fatty acid synthesis complex. Also makes it difficult to work with them because it's this complex. You have to have a whole bunch of proteins you put together. Hard to work on one of these in isolation. Okay. Fatty acid synthesis uses 2-NADPH, while beta oxidation yields yields 1-NADH and 1-ubiquinol. I'm going to write it. 1. All right, and from my perspective, when I think about this, I'm treating that NADPH and that NADH as energetically equivalent, right? So they're not the same molecule, but energetically they are. And then we can also say fatty acid synthesis, or let's say in fatty acid synthesis, the growing chain is covalently attached to a protein. To an acyl carrier protein. Okay. In beta oxidation, the fatty acid is attached to, what's it attached to? Covalently, a CoA.
Oh.
And I'm going to add covalently attached to a CoA. I think that's everything. I'm going to scroll up real quick. Yeah.
Is this all done in the cytosol?
Yes. Yeah. That's a good other difference. Let's talk about that. So fatty acid synthesis occurs in the cytosol. And beta oxidation occurs in the mitochondria. Okay, and that's important as we talked about before for regulation. I've got a couple practice regulation problems at the end of the lecture. I don't know whether we're going to get to them. They may be next time. Yes?
Are the fatty acids, whenever they're like synthesized, are they just made and then shipped somewhere, or are they made on the structural protein, whatever they're doing?
They're made on the on where they're going to be, like for the
function is.
I don't know of an example where a fatty acid is actually synthesized or a mechanism by where it's synthesized on the actual protein it's going to be made. So it would have, and I think it probably has to be made in the acyl carrier protein complex and then transferred. I don't know the mechanism of transfer for that. That's not a pathway I've seen, but there's got to be one, right? In terms of when we talk about protein synthesis, we'll briefly touch on how the carbohydrates get made, and they actually get built onto the protein often. Sometimes they'll be built on something else and transferred over, but we'll talk about that. Good question. Okay, other questions before I move on? All right, we're still talking about the same thing. So I want to talk about biotin. How many people have heard of biotin? It's actually, what vitamin is it? Anybody know? I've forgotten. It's a vitamin something or other. It's what? Okay.
There's only one.
So biotin's going to be used. It's a B something, but biotin is used to actually add that carbon dioxide. So when you need to add a carbon dioxide to something, biotin is the cofactor that gets used. So this biotin, and the biotin, when we think about the process by which this might happen, this is going to be attached to a lysine. Okay, so you end up with attachment to this to a lysine. So it's actually, well, yeah, I did. Let me go ahead and cross that out. I'm going to attach it to the lysine. It was a carboxylic acid. So what kind of bond are we probably making? Yeah, an amide bond, a peptide bond. You could think about it as. So this is biotin is going to be attached in your enzymes. Okay, like your FADH2 is. It's not covalently attached, but it's held really tightly. So we covalently bond this. Oh, it's more like your lipolysine, right? Lipolysine. Okay, I would like to call it lipolate sometimes. So this is going to be attached to an enzyme, and then it's going to, we'll see in this mechanism, it's going to carry a carbon dioxide on it. So what we're going to have for this is we're going to have a recognize, draw the other form, loaded or unloaded.
What is this in relation to?
This is how we're going to put a CO2 on our malonyl-CoA. Okay. Also, remember for gluconeogenesis, we take pyruvate, we make oxaloacetate by putting a carbon dioxide on, and then we knock it immediately off. Biotin is involved there as well. This is, what was I going to write? This biotin is the cofactor that is involved in the addition of carbon dioxide to molecules, we'll say. Okay, and so where we've seen it before, we didn't talk about biotin before. But we can go with examples. Making of malonyl-CoA. And, oh, I forgot now. Making of oxaloacetate from pyruvate. Okay, we're going to have this cofactor involved. Let's see, do I get a picture of where it is in the biotin? I do. Okay, so we'll see down below where we attach to the biotin with the carbon dioxide. Another interesting thing, which I won't test you on, but I have to talk about anyway, is biotin. This is actually in molecular biology, this is a really useful molecule. It can be bound very tightly by a protein called streptavidin. So you can have streptavidin binding to biotin. It binds super tightly. It binds at about 20 kilocalories per mole of affinity, and that doesn't mean a lot to you, but it is kind of the record holder. So, you know, I'm down at BT and we're talking about how good our calculations are or things, and we get something that binds tighter than 20 kilocalories per mole, we know our stuff's kind of off, right? And it actually, the stuff we use actually is off. It's always binding too tightly. But yes, that's the record holder. If you want to see whether you think you believe a result or not for binding, and if it's at negative 40 kilocalories per mole, you don't believe it. Okay. But so it is super 20. It's the record holder. So people will use this. You'll take the avidin, you'll take a biotin, you'll stick biotin on DNA, and then you can have streptavidin with a fluorescent label and use that to label the DNA. You can do all sorts of fun stuff, and people do. I used this when I was in industry. Okay, let's talk about making the malonyl-CoA. We've only got 3 minutes left here. Question?
You told us to remind you at 3 minutes.
Oh yeah, well, we're going to talk about this first anyway. Thank you, though. I appreciate it. All right, so let's just quick look through this mechanism. You should be comfortable with this mechanism, okay? Be able to draw it. But what we've got, or draw a step in it at least. So if I was to give you arrows, so what we've got here is we've got an acetyl group here, right? What we can do is activate that acetyl group as carbon dioxide, right? Take this carbon dioxide in the form of bicarbonate, it can attack the phosphate on the ATP. This is where our ATP gets used. We get a phosphate on there, and now we have a good leaving group, and our biotin can attack there. And then we have our biotin has this activated carbon dioxide on here, which can be used to add to a growing acetyl group. We could take the acetyl group on the acetyl-CoA and add that there. Okay. This is called making malonyl-CoA from acetyl-CoA. So we'll call that the mechanism of making malonyl-CoA from acetyl-CoA. Okay? So know that. Be comfortable, you could fill in a step of that, or that you could discuss why this, why do you need that? Why do you need the ATP to make that? Okay, let me go ahead and hand your test back. Your test, so I curved the test by adding 5 points to everybody's score. It's not reflected on your score on your test, but it's reflected in the grade book. All right, so check my math. If your score in the grade book is not 5 points higher than what is on this test right here, let me know. The answer key is posted. I encourage you to look at it and then come ask me if you have questions. But if your timing is such that you just want to ask me questions, I'm open to that as well. I don't want to discourage you from asking questions. And then once you have your test, you're welcome to stomp out or whatever you like.
She would also have like one of them, just like when people want to make that pictures, like I know.
Yeah.
Zero.
Remember, I have office hours, 1:15 to 3:15 today. Happy to see people then.
Yeah, I said.
I can't remember. Is Reese's 3:6?
Yeah. I've done that books. You can see like what I like struggle with.
I like to take the pictures so
Yes,
that I had
yes.
the same exact one.
Yes.
They're not there yet, but that's all right.
I'll get them up.
If I like,
Depends on how busy I am this afternoon. Try to get them up right away.